Yeah, so as I was saying that we learned today a lot about the application of structure, the application of chemoinformatics for biomedical drug discovery. So in this entire program, we will be going and learning about different aspects of biomedical drug discovery. So we started with genomics and we wanted to learn about the sequences and the differences in the context of a disease. And then we learned about the gene expression in the transcriptomics program. And also now we are going to learn about the application of data science and how we are using data science in this entire process of biomedical drug discovery. And alongside with the data science and all of that sequence-based knowledge, now we are transitioning into the structural space. So it's very exciting to be able to do this with all of you here who would be joining us for this entire course. This will be an intensive 1 month course where you will have like regular sessions to discuss and where we will be going through the topics that you saw that Dr. Sujit showed you that will be covered during this period. And also, uh, the purpose of doing this OmicsLogic training programs uh, is to help you and guide you develop these, learn from these techniques and develop research, research ideas, research projects. So one of the purpose would be to learn from the very basics. So that's why there are two phases in this program. The first phase focuses on the technology and the techniques that are very much used in today's biomedical drug discovery pipelines. So I think many of you have heard about protein structures, and with the SARS-CoV-2, the variants and how the variants change the structural, the function and all of that. So I think we will be covering a lot of those basic concepts of how do you understand, how do you visualize, how can you look at the structure. So that's very important, because in the end what we are trying to find is we are trying to find different strategies to be able to understand and devise using these different tools. But again, we will have to go back to the very basics of understanding these 3D structures in quite detail, so that we can apply these different techniques like QSR or docking or molecular dynamics. So in this program, we will be covering some of these techniques which will be used in the context of a disease. So if you are already working and if you are a full program participant, then we will be working in for a project that could be applied for these different diseases. If you already are working on a project, so many of you are researchers here and are working on different sort of research topics or labs where you are working on different projects that are related, somehow related to biological research. So in that case, if you're working on structural data, if you're working on sequence data, if you're working on a protein, if you're working on some drug discovery aspect, then it's a program for you to join. Because in this program you can reach out to us. We will be there to guide all of the students who would be joining to devise the strategies, and what are the strategies that you can take forward to to help your research go to the next level. So if you are working on some hypothesis, you have some goals, some research objectives that you have defined in your thesis, maybe that you have defined in your in the next few days that you will be covering. So all of those will be there. We'll be helping you with that. So as I was telling you, the next session would be will be a free session, which will be an intensive session because it's going to start at 6 p.m. First, we will have the Schrodinger workshop for 1 hour, where you will get an overview of the entire software and what are the different applications. So as I was telling you, that OmicsLogic learning helps in just not about learning, it's about application and learning the logic. So we will be using two proprietary softwares here. One is the TBioInfo platform for most of our analysis that we did in for genomics, transcriptomics, data science, and also there are aspects of data science and chemical space structures, two-dimensional or three-dimensional structures on molecules that will be used for machine learning. But other than that, we will be using two proprietary softwares. One is the TBioInfo platform and also the Schrodinger platform. But the logic of the analysis would be same for every kind of research that you will be doing. So you can apply those techniques and use open source softwares in the next stage, where you are trying to do your own research, and you not to be dependent on it, but to be learning the logic and how to apply these techniques and get an outcome or a result that actually relates to the biology of the problem. So how close you are to the problem of mimicking this uh, in biomedical drug discovery. So we will be doing docking simulations. So we will be doing learning about molecular dynamic simulations. So what we are doing in those simulations is to trying to mimic the stages where we can try to see that how the molecule fits, if it is stable or not, what would be the free energy. So all of those will be covered in the program, in the context of the program, when we will be having the workshops. And then we will have the understanding of how computer-aided drug discovery is useful in this entire process of biomedical drug discovery. So we have another session after that on 1st July by Dr. Chakra Mesha, and you've already heard about it. Like, it would be about target identification and validation. So how do we identify targets at the beginning of the experiment? And if you already have a target, then how do you validate that to be a good target? Whether if you find if you're looking for a solution, for example, if you're looking at a disease, if you're looking at want to work on a specific disease. So we have different examples here in our OmicsLogic training, where we have gone through infectious diseases. So we have gone through examples of COVID-19, malaria, and tuberculosis, all of those examples to learn from. And then we have examples in cancer research, where biomedical data drug discovery and machine learning and using patient information, all of that is much used, and there's a huge application. So understanding that in the context, and how to apply this in using programming languages such as, or statistical languages such as R. So there will be another expert session by Dr. Shailu from AIMS on 14th July. That would be about cheminformatics using R. And then, as you already have seen, that understanding the recent advances in drug discovery. So as I was telling you, that machine learning, first we learned about these techniques, then we discussed about the way it is being used in drug discovery. But in cheminformatics, like I think artificial intelligence has inspired this computational aided drug discovery, and this widespread adoption of machine learning. In particularly, you have heard about deep learning, you have heard about the Google platform that came up to predict uh, protein structures. So understanding the way modern drug discovery is working, how artificial intelligence is enabling this entire process of drug discovery, and it's fueling the development, right? So there has been a kind of skepticism before when we started with all of this, but this growing data and the continuous development of these resources have really led to this entire process to be really much more defined, and now we are believing in it and we are seeing some results. So like, this benefit comes from medicinal chemistry, comes from AI, comes from all of those allied fields, radiology, and we are using patient data, so EMR records and all of that. So understanding drug discovery in this entire intensive program would be about multidisciplinary aspects of drug discovery. So it will not be just about finding a molecule and docking and just finding the structure, but to understand in the aspects of the disease, and if there are resistance, how to overcome that. If there is a problem of drug repurposing, how to look at that, and how to incorporate multi-omics data as well as cheminformatics information into a project. So these are some of the objectives that we have started with, and I believe that we will be able to complete some of these analysis during this program and help you all to learn from what we have gone through in our entire career, what we have learned from all the experts. And we have a group of experts from a lot of experience from what they have gone through in their own journey in pharma. So I'm really excited to be a part of this program and become and be a mentor to many of you who would be working with us on these different aspects. Now I'd like to hand it over to Sonalika to tell you about the registration process and how can you come on board and start working with us. Thank you.